Hello, I am Dr. Kuzma Strelnikov from Toulouse University Hospital. I study the brain, and in this video, I would like to discuss a rather general question about brain function. What is brain activation? This question is pertinent to a branch of neuroscience called cognitive neuroimaging, which studies brain activations in response to cognitive acts, cognitive loads, cues, and so on. So, if we study brain activations, there is a legitimate and, in fact, rather basic question. What is brain activation? Why do we see blobs of different intensities made up of points with varying intensities? In functional images of the brain, like fMRI or PET scans, I became interested in this question after my PhD. And interestingly, I couldn't find any satisfactory answer in the literature, in scientific literature. So, in the end, I published my own definition of brain activation. And in this article, I defined brain activation as information driven reorganization of energy flows in and among populations of neoglial units, leading to an overall increase in energy utilization in these populations. This definition needs some clarification. Neoglial units are groups of neural and glial cells in the brain that work together. Energy flows are transformations of energy that travel along the brain's structures, like axons, synapses, and so on. In these flows, metabolic energy is converted into the energy of electromagnetic fields, and when at rest, without any stimulation, the organization of this system of energy flows reflects predictive coding, and the system is reorganized afterward by stimulation. Predictive coding can be clearly illustrated by our study of patients who were initially deaf and received cochlear implants. At rest, without any stimulation, they showed differences in activity compared to controls in visual, auditory, temporal, and speech-related areas. In all these areas, there was an increase in activity observed without stimulation 6 months after their implantation. Thus, these areas support the increased predictive coding for audiovisual language that these patients need. In schematic terms, the relationship between informational inputs and predictive coding can be clarified like this. When there is a large difference between the received information and our predictions, we need much more energy to process it. When our predictions match the received information, almost no additional energy is needed compared to the resting state. Energy usage needs to be economized or optimized in the brain. This is why we have specialized cellular populations in different parts of the brain that can predict various types of incoming information, such as auditory or visual information. The proposed definition refers to the unity between the two types of brain cells, neurons and glia. These neuroglial populations exist in 3D, in 3 dimensions in the brain, but for computational purposes, can we represent these populations simply as cubes? Of course, this is a serious idea. Neoglial populations have different sizes and shapes in the brain. However, interestingly, we don't have a choice in neuroimaging. Any neuroimaging technique represents the brain as a set of small cubes, called voxels, just like pixels in pictures. We have a representation of the brain as a 3D activity, where each cube has a signal value which reflects the average level of metabolic energy in that cube. What happens when a signal arrives from one neoglial population to another? Electrophysiological studies show a burst of activity at the site where the signal arrives, corresponding to a steep increase in energy at that location. For a curve in one dimension, the slope is mathematically defined as the derivative of the curve. In multiple dimensions, the slope is mathematically defined by the gradient vector, which points in the direction of the greatest increase of the signal. If we're lucky enough to divide the brain into voxels, small cubes, pretty well, it is quite possible that we might see a sudden burst in one specific voxel and yet observe absolutely no burst at all in the immediately neighboring voxel. And this results in a high gradient between these voxels. For example, in the diagram, if a voxel has a signal level of 3, the greatest difference will be with the voxel that has a signal level of 8. This illustration of a single slice of brain activity shows that gradient vectors can create divergences, spreading out from a source, as well as convergences. We ask whether it's possible, by calculating gradient vectors in three-dimensional brain activity, to identify statistically significant and stable activity flows, especially those specific to a particular type of cognitive load unique for each kind of cognitive task. For example, one might expect to find sources in the occipital cortex, in the visual cortex, for face perception, and then see the propagation of activity into the frontal cortex. After analyzing two groups of subjects, one exposed to visual stimuli and the other to auditory stimuli, where the visual stimuli involved faces and the auditory stimuli involved words referring to those faces, in fact, there is a statistically significant observation of sources in the occipital cortex for faces and in the temporal auditory cortex for auditory words. We also found significant directions of stable activity flows, for example, in the forward-backward direction for faces, which corresponds to the y-axis of the brain. In another study, we found a high spatial overlap between the gradients measured in the brain. Using fMRI, which reflects the brain's metabolic activity, and using EEG and MEG, which reflect the brain's electrical activity, these results were obtained with exactly the same stimulation. This suggests that the observed gradients in brain activity are not just a result of one neuroimaging technique, but truly reflect how the brain functions. Therefore, our results indicate that there is stimulus-specific spatial organization within the activated regions of the brain. And this spatial organization encodes the presented stimulus. And finally, it forms spatial patterns of brain activity, or spatial patterns of voxels in the brain. From a fundamental perspective, spatial coding can be appreciated at the intersection of the three main physical properties or phenomena: information, energy, and space. One might ask, if spatial coding exists within brain areas, can it be shared between different brain areas for a given stimulus? For example, during face perception? One could expect similar relations between voxels in the occipital areas, in basal, temporal, and frontal areas, known to be active during face processing. We'll discuss this interesting question in another video. Thank you for your attention.